to Biostat Squid. In this tutorial we will learn how to create and customize a heat map to visualize your differential gene expression analysis results. We will use the R package pheatmap, which gives us great flexibility to add annotations to the rows and columns, and we will also cover other ways of customizing the plot. If you're not familiar with heat maps, you might want to check out my other video where I explain how and when to use heat maps to visualize your data. And as always, you can find the code I am using in this tutorial at biostatsquid.com, where you can also find a step-by-step explanation of the code. For this tutorial you will need R or Rstudio, and you will need to install the pheatmap package. So if you're ready, let's dive in. Okay, so this is our script, and for those of you who already watched some of my other tutorials, you know I like to clean my environment up in case we have hidden objects in the background, free up memory, and then set our working directory. So first we'll start by loading our relevant libraries. I load the tidyverse. It's not really necessary for this tutorial. I just like to have it there for data manipulation, but I don't think we will need it. Then we have the library pheatmap. This package is very important. This is the main package we will use today to build our heat map. And then our color brewer I will just use to get colors and color palettes for my plots, but it's not necessary. You can use other packages out there for your colors or just input them manually basically. So let's start by setting our input path. This will also be my output path, so this is where I have my data and where I will output my plots. Okay. So now that we have our environment set, we'll start by inputting our data. And in this case, I am not actually reading in a table like you may have, but I will create my test data from start just for demonstration purposes. But in your case, you would maybe use a function like read CSV or read table just to read your data in. So these are just steps to create my data frame and give names to the columns and the rows. So if I run head, we can see that there are different columns with our sample names in this case, and then different rows with our gene names. We can also have a look here. So this is what your data frame should look like. So each column should be a different variable, so either patient IDs or cell line IDs or samples, and each row should be a different gene, or if you've done pathway enrichment analysis, you might have different pathways in each of your rows. Great. So now we will actually try and get a heat map. And with the pheatmap package, it is as easy as calling pheatmap. So if we run this already gives us quite a nice heat map. You can zoom in to observe it. So great. This is basically it. And now we're just going to go over some ways of customizing this plot. So you can basically customize it to whichever way you want to display the data. So first we'll talk about clustering. In my previous video I also go a bit into clustering, and in this first heat map we already see that the columns and rows were clustered. But you can actually customize this clustering methods using cluster rows. So you can set it to true. It's set to true by default, but anyway, and you can cluster your columns by setting it to true. If you don't want to cluster your columns or rows, then you would just set it to false. Then we have different methods. I won't go into it in this video, but you have different methods to cluster your rows and columns. I will set both to Euclidean. I don't want to misspell that. Okay, and also my rows. Let's see, and the clustering method I will set to word. You can actually test different methods to see which one groups your rows or columns better. So this is it. We get a very similar heat map. You can barely notice any difference basically because the heat map was already clustered before, but there might be bigger changes depending on the parameters you set there. Great, so now let's customize our heat map, which is the fun part. Right, so first step might be to add a title, and that is as easy as calling main. So main is the main title for the heat map, so I'm just going to call it super cool heat map. And there it is. This is my title. Of course, you can also control how big the font is and so on. Right, and you might want to decide whether you want to display your rows or columns. So I'm just going to copy paste this. Let's see, and perhaps now I want to show my column names, so the sample names. If I don't want to display them, I can set it to false. So full stop. Okay. So here my column names disappeared. I will set it to true because I want my sample names to be there. Also show row names. Again, these are set to true by default, but you might want to change that. Then I can actually display the color of the values, for example. Give me a second. So font size number equals 4, and the number of the color number I will set to black. Okay, so this is if we decide to display our numbers, and if we set it to true, it displays our numbers. For example, see? And I set the font size number to 4, but you can actually change it and make it bigger, for example. So this is if you actually want to show the values of your gene expression data, basically. Great. You can also, well, this should actually be down here in this section, so showing your values. So I'll just copy paste that there. Okay. So you might also want to change the color of your cells or the border color. That's also customizable. Let's see. So you would do that by setting the color parameter, so col. And here I will use a brewer palette from my library, or color brewer. So for example, I will select 10 colors from this color palette, which goes from red to yellow to green. There are many different palettes, so you can actually visit the webpage and it will, it will tell you everything and you can choose them from there. By the way, you can also change the color of the border with border color, and set it to black, for example, or to blue, or whichever color you want. I think the default is a type of gray. So now we have some more colors. So this is my color palette. It goes from red to yellow to green. And here I'm displaying the numbers. I can remove that if I want. For example, display numbers equals false, and now they disappeared. So it's a really easy way of customizing your heat map. Nice. So you might want to customize your legend as well, and that's quite easy. If you can guess. Well, by the way, if you want to know how to do something, you can of course call the help function. I'll just write it here, pheatmap, and if you run this, it should give you all the arguments you can pass to this function. Of course, you can also Google it, and you might get faster answers. But yeah, there's basically anything you want you can do with pheatmap. Right, so where were we? Legend customization. So let's set the legend breaks to -2, -2, 0, and 2. These are just my legend breaks, so where I want to actually show my the numbers I want on my legend regarding the distribution of all gene expression data. So you might have, I don't know, gene expression data that goes from -20 to +50, and in that case you can set the breaks, so which numbers you want to display on the legend, if that makes sense. In this case, I don't want, well, I'll just run this to show you. So see, now I'm displaying -2, 0, and 2. But perhaps those are the values, the breaks are the values I want to display, but perhaps I want different labels. So I can set different labels. So in this case, I will set it to low expression, medium expression, and high expression. And there you go, low, medium, and high. So that's also quite a nice way to display a legend. You can actually just set the legend to false. And if it's set to false, we don't have any legend anymore. You can, of course, also decide where in the plot your legend goes, the legend location. Right, so another cool thing you can do with pheatmap is actually split your clusters. So you would maybe want to do this to highlight certain clusters. So for example, you might want to divide up this cluster over here and separate it a bit more from these two clusters over here. Or perhaps you have a certain number of genes, perhaps we can zoom this up, perhaps you have a certain number of genes up here that belong to a certain pathway, and you just want to highlight that they're different from the rest of the genes. And you can do this quite easily by calling heatmap data, there you go, cut tree rows. So for example, we want to have 2 distinct clusters and columns. So say we want 3 of them. So if we run this, there you go. So now our heat map is displays 3 sample clusters a bit more differently, or more, they're just more highlighted. See, there's a slight separation between them, and also for the rows. You can also just do it for one of them, of course. Nice. So what else? Another thing you can do to customize your heat map is change the font size of your columns, your column labels. So I'm going to set this to 7, and then the rows I will set to 10. So if we now run this, see the sample font size is now smaller, and also the gene or the row label size. This is not very nice. For example, you might want to change the angle, so you can do this quite easily with the angle call and set it to 45. Let's see. So now, whoopsie, my plots. So now they're much more readable, basically. You can, that's about it. You can also change the border color and set it to false if you don't want a border. You might prefer this kind of look. You can also highlight the missing values with a different color, with any color, and set it to black. In this case, we don't see any changes because we don't have missing values in our data frame. But yeah, basically anything you want to customize, you can do it quite easily with pheatmap. Right, so now for our last bit, let's talk about annotations. So perhaps you want to display different annotations both for your samples or for your observations, so your genes basically. And you might want to import your own metadata, so your own column or row annotations. I will just create it easily enough with this couple of lines, and I'll just show you and data frame. So this is the annotation data frame for my samples. So it's very important that the row names are set to the sample names. So I did this with this line over here. So each row name, sample 1, sample 2, sample 3, should be the row name of a sample basically. And then you have here, each column is basically the metadata I want to display. So in this case, I added a group. So I divided my samples into disease samples and control samples. And then I also added another column called lymphocyte count, which has the number of lymphocytes, whatever that means, for each of the samples. So you can have categorical and numerical data. You can have as many columns as you want. Obviously, you don't want to have your heat map too overcrowded, but anyway. And I also wanted to show you your gene functions, which is already here, a bit of a spoiler. So I will create a data frame for my gene annotations. And in this case, I will just add a column called gene functions, and the same thing. Each row should be the name of your genes, of your observations, and that's the row names of my data frame, of my annotation data frame. And then in this case, I only have one column called gene functions, and I have different functions. Of course, you can have your own annotations. Just make sure that the row names are set properly. Nice. So once we have this, we can also create another dictionary or list basically to set our colors. This is not necessary. I just want to customize the colors I give to each of my annotations. So it looks like this. So this is a big list, and my list is made up of basically it's kind of like a dictionary. So for gene functions, so this would be the title. Where is it? I lost it. So this would be the name, the column name basically of my gene annotations. You have to make sure that you have all the possible values or categories for that annotation. So I have oxidative phosphorylation, cell cycle, and so on. And I assigned a color, and you can use the hexadecimal color code, or you can use names. There's many ways of doing this basically. Then I also have my group and lymphocyte count. So basically, and colors is just a big list, a big dictionary, which has colors for each of my annotations. This is another shortcut. So I use the Brewer palette to give me, I will just run this so you can see, to give me 5 colors from this palette. So I will just run this. So now I have my colors list. Nice. So let's create an annotated heat map. So again, really easy with pheatmap. I will just keep my previous color palette for all the values, gene expression values. Nice. Right, so to add annotations, we just use annotation row. So for annotation row, I will use my gene functions data frame. For annotation col, so for the columns annotations, I will use my data frame. Of course, you can use different names. Nice. And for the colors, we cannot forget about the colors. Annotation colors. I will use my dictionary. So colors. This one here. Very nice. And let's add a title. Super heat map with annotations. Smiley face. Let's run this. See if it worked. There you go. So this is a very nice heat map. It has my annotations. As you can see, maybe there's a bit too much color here, but anyway, this is just for demonstration purposes. So you have your lymphocyte count, which is a gradient, perhaps. Then you have the season control. I set it to green and purple, and then different colors for our gene functions. And this is quite a nice way of customizing the color of your annotations, because you might change. So as your analysis goes on, you might decide to color oxidative phosphorylation in a different color, and this way you just have to change one line in your annotation colors dictionary, and it will apply to the whole heat map basically. Nice. So that's basically it. So let's put it all together. So this is the final heat map I wanted. So you have data, you have the color, the clustering, the annotations, the font size, whatever. So I will just run this bit here just to show you what my final heat map looks like. So this is what my final heat map looks like. And I'm happy with this. And now I will assign it to this object called heat plot. So now, as you can see here, this is heat plot, and I think it's a ggplot object. No, not sure. Anyways, it's an object. Let's leave it at that. And that's my object. And now I want to save it. So I can save it to a PNG. I can save it to PDF. In this case, I will save it to a PDF. I will set the height and the width. You can also set the title of it, the title of the file. This is just where I want to save it at. So using paste 0, this, if I run this, it basically prints something really scary. Let me see, my path is set to okay, and then, oops, yeah. What I did wrong was having this here. So I want to save my heat plot. Actually, what you do in these cases, you just call PDF. So first we need the name of the file. So with paste 0, I can give it a name quite easily. Heatmap test .pdf. And if I use the paste 0 and the path, then it will be saved here. So this is the final file name and location. Then I have to say what I want to save. So that's here. And height and width. Nice. So I will just run this. And there you go. Now I saved my PDF to this folder. Nice. So thank you very much for watching. If you're new here, don't forget to subscribe. Let me know what you thought of this tutorial. I really hope it was helpful, and I really appreciate all your comments and support. Have a squid-tastic day and see you in the next one.